My last name is a lot easier to say, so go by Chu, as in Chewbacca or Pikachu. So it's a lot easier to remember as well. I lead the high-throughput biology team here at Xaira, and happy to share with you our efforts in the virtual cell work. But before I start, maybe just a couple quick words about the company. The company name is pronounced Xaira Therapeutics. It may be as confusing as my first name. So think Xaira Thera. We are a AI drug discovery company. What that means is that we are going to make drugs to advance patient care. So we're a fully integrated biotech company, but we are also an AI company. We're innovating on a few AI platforms that will help accelerate and improve the drug discovery process. That involves not only vertical expertise in therapeutic discovery, which we have a lot of those talents in the company, but also involve frontier AI as well as massive data generation. As a company, we believe that biological data are fundamentally limiting. We don't have the internet text that's available for training consumer AI models. A lot of this data still have to be generated ourselves, so we believe in investing in both data generation as well as AI model building. And where are we building these AI tools? Well, maybe I can direct your attention to the left of this figure. We like to say making a drug is as easy as 3 steps: finding the right target for a disease, making the right molecule to engage that target, either inhibit it or activate it, and lastly, recruiting the right patients that we believe will benefit from this treatment. If you do all 3 steps right, you will have a successful drug. But of course, in practice, each of these steps is extremely difficult to do right. Traditionally, this really relied on a lot of empirical trial and error. It's an artisanal process. Every step has incredible attrition and overall leading to the low efficiency of the drug discovery process. On the right, we're now approaching this era of AI-enabled drug discovery, where we can build tools, AI tools, to accelerate and improve the success rate for each of these 3 steps. So on targets, which will be the bulk of the talk today, we're thinking about building a virtual cell that can truly causally map biology, predict what's going to happen to biology if we did certain perturbations to the cell. That'll give us better targets we haven't thought about before. On design, on molecules, we're applying the latest molecular design AI tools so that we don't have to go through expensive and difficult screening efforts. We can design these molecules in silico and have a higher success rate, even go after targets that weren't previously possible before, so-called undruggable targets. Last but not least, a more nascent effort in the company is to collect patient data and building patient models that can stratify responders from non-responders and guide trial design. And so throughout all of these, we hope to turn drug discovery from an artisanal process into a in silico or a engineering discipline. And threading all of these two together, we're also building agentic AI, which we call X scientists in the company, because everything starts with an X at Xaira. X scientist agentic AI to integrate the deployment of these tools and make it available for all of the biologists, not just the AI experts to use. So with that, I probably only have time today to talk about the virtual cell effort, and I'll go into this section now. And before I start, just for folks who are maybe less familiar with the space, I like to draw an analogy to a large language model training that we're all very familiar with this by now in the consumer AI space. In consumer AI, we've all seen these large language models being pre-trained on large amounts of text data, text corpora data curated from the internet, for example. And by training the next token prediction over large amounts of data, these models learn the syntax, grammar, and knowledge of the human language. And these models can then be fine-tuned using specific data sets through, for example, human reinforcement learning to become helpful tools for specific applications, such as a chatbot. Importantly, the training data is quite appropriate for the task at hand. We're seeing a very similar paradigm in biology models, where we see these architectures actually very similar to GPT, for example, SCGPT or single-cell GPT that was pioneered by my partner in crime, Dr. Bowen, when he was still at University of Toronto. Training single-cell RNA-seq data that's curated by cell-by-gene effort from CCI, 33 million single-cell RNA-seq data, and follow a very similar training paradigm, next token prediction or next gene prediction. These models are able to learn the syntax or the language of single-cell RNA-seq data. And these models can then be fine-tuned using patient data, for example, to become helpful tools for patient stratification, task-specific applications. But importantly, I just want to stress, the early generation of these models are all trained on what I call descriptive data. So these are profiling data sets, mostly curated from healthy human donors. There's very little perturbation, very little cause and effect in these data. And so what we see is that these models perform quite well on descriptive tasks. For example, cell type annotation, batch effect normalization. They don't do super well yet on perturbative tasks or counterfactual questions. If I did this to the cell, what would happen? And one might argue that these counterfactual perturbation prediction is more useful for drug discovery, because it enables the ability for us to do in silico perturbation, in silico prediction. I would argue that a key missing ingredient in this whole training recipe, this analogy, is that perturbative data is missing in training. And if you don't have causal data, of course, the model is ill-equipped to learn what is causality in biology. If you were to ask me, how do I break down the different training data that will be useful for building the virtual cell, I would say, of course, we still need a lot of descriptive data, cell-by-gene efforts, for example. We also need a lot of perturbative data, cause and effect, perturbation and outcome, and that will be the bulk of the talk today. We think this will truly teach the model about causality. We also need a lot of translational data. These are data that you gather from high-quality preclinical models like animals models, organoid models, for example, as well as from relevant patient tissues to really ground an in vitro-derived model in patient reality. Perturbative data has been sorely missing in this field. And so we saw a gap and opportunity, quite frankly, to generate perturbative data at scale to really infuse causality into model training. And I already discussed many of these aspects. Our paradigm for building these virtual cell model, which we call XCell at Xaira, follows 3 steps. You do causal learning by generating massive causal data and training a model. You then demonstrate model capabilities, both in forward prediction and reverse prediction. Forward prediction being, if I did this to the cell, what would happen? Reverse prediction being, if I want to move a cell to this particular cell state, what perturbation do I have to introduce? If a model can do both forward and reverse predictions well, it will be able to answer a couple key questions in translational applications. For example, what targets can move my sick cell towards a healthy phenotype? So this is the what question for target discovery. But not only the what, if we marry this model with a structured design model, we would also hopefully be able to answer the where question. Where on the target protein should I intervene that can give us the desired phenotype? Because the model is expressive, we can infer what's going to happen at a molecular state upon the perturbation. It should also be able to answer the how question. What is the mechanism through which this perturbation is worsening or improving the disease? And last but not least, when matched to the right patient data, a well-trained foundation model hopefully should be able to answer the who question. How are these patients different from each other and who is going to benefit from a particular treatment? Another important aspect of the model that we hope to achieve is this idea of context generalization. And that is because biology, just like any consumer AI domain, there are data-rich regimes and there are data-poor regimes. Data-rich regimes are cases where we can generate massive causal data at scale, for example, simple cell culture systems. Data-poor regimes are systems that are not amenable for high-throughput experimentation, for example, animals, very complex multicellular systems, and human explant tissues, where you hopefully can generate just enough data to fine-tune the model and leverage the massive causal network that's learned from these simpler systems and transfer the causality to systems where you cannot do exhaustive experimentation and you can rely on the virtual cell model as an oracle to propose the right experiments and test them with high success rate. And if a model can achieve this data-rich to data-poor regime transfer, I think that's really the holy grail for building a virtual cell. So how do we generate these data, causal data for model training? Well, there are many different ways. In my previous life, working at In Sitro, another AI drug discovery company, we spent a lot of effort building a pooled optical screening platform, which we call POSH. And the team figured out how to make POSH compatible with high content imaging, for example, cell painting. So shown here are cell painting, which is a multi-stain panel staining different organelles or macromolecules in the cell, followed by rounds and rounds of in situ sequencing to figure out the CRISPR barcode that's present in every cell, thereby tying the genotype, the perturbation identity, to the phenotype of the cell. And three innovations that we introduced at In Sitro is full end-to-end work cell automation. So we completely made this 24/7 work cell that can generate these data at scale without human intervention. Deep learning analyses, embedding these high content imaging phenotypes through a deep learning network that we actually here we borrowed DynoVIT open source model from Meta that are used to train cats and dog, and segmentation, consumer AI image analyses, which work surprisingly well on cellular images as well when you train a model on our internal data. And the output of this is that we achieve much higher sensitivity in detecting perturbational phenotypes than the traditional image visualization engine, for example, Cell Profiler. We almost triple the sensitivity of phenotypic detection, and the embedding achieved by these deep learning networks are much more semantically meaningful and rediscovering known gene-gene interaction, for example, that's curated by StringDB databases. So this work is published end of last year. It shows that we can use high content imaging paired with high throughput CRISPR perturbation to learn gene function. The technique that we have focused on here at Xaira is quite a bit different. Here we use a technique called CRISPR called Perturb-seq that marries high throughput CRISPR perturbation in a pooled fashion with single-cell RNA-seq, which produce these rich matrices or tensors of data which have high throughput perturbations on one axis and high dimensional gene expression assays on the other axis. Now, this technique, we stand on the shoulder of giants. This technique has been pioneered in academia by Jonathan Weissman, Avi Regev, Chris Bach, and colleagues. Even genome scale Perturb-seq has been achieved before. However, this is very difficult to do routinely at high throughput. When we are doing large-scale genome scale experiments, we're talking about harvesting incubator full of cells, put them through FACS sorting to enrich for the cells that we want that express CRISPR. Often that process can take upwards of 16-hour days. And traditional methods rely on fresh cells for cell handling. You can imagine fresh cells that go through that long day of process. By the end of the analysis, you can readily detect stress signals in your cells as well as in your scientists in the data set. And that's all of the artifacts that we'll be putting into model training. And very early on, we realized this is not a way to generate AI quality data. So our scientists actually put a lot of effort into developing fixation methods, cryopreservation methods, so that we can fix the molecular state of the cell and render them immune to downstream human handling. We introduce QC stop points, which I'll talk about more before we go into large-scale data production. We automate the library steps, and the end result is that we're able to generate superior data quality compared to that, even the benchmark data from the Weizmann lab, the Roplogo Perturb-seq data sets. And just to go through some of these optimization steps, we figured out that the best fixation agent that we could find at the time is called DSP. This reagent can cross-link the cell, but is readily reversible upon reverse transcription because of the reducing condition that's happened in those reactions. And so it is introducing minimal signal dampening or loss of sensitivity in RNA-seq. And the the end result is highly correlated to that of unfixed cells. We also had automated library prep steps. And this is important because for small-scale perturb-seq, human manual library preparation using a 10x genomics platform completely works. But when we're talking about genome scale preparations, often we're talking about many tens, if not hundreds, of library preparations. And here automation really helps. And our team worked through every step of the library prep and really build a robust workflow that achieved very low CV, in many cases less than 1% CV, and produces library traces that are indistinguishable from manual processes. And this made our entire data production pipeline much more robust. And so just to summarize some of the optimization steps, the fixation made the RNA immune to degradation, and we're able to get much higher sensitivity of data, higher than that of the reportable data set. The pilot QC was another important introduction to the workflow. We don't have to process all of the samples in one go. We can have a pause point, process a tiny sample from our entire genome scale screen, be able to show that these data are good before we launch a full genome scale perturb-seq experiment that costs hundreds of thousands of dollars and at a point of no return. We were able to superload our fluidic systems up to 5x, and that really reduced the number of batches that we have to run and introduce further cost savings to our workflow. And last but not least, automation was able to let us do hundreds of library prep while producing very similar results manual steps. The end result is that we're able to achieve really state-of-the-art sensitivity for single-cell RNA-seq, and really preserve long-term, achieve long-term cryopreservation in the freezers. We were quite surprised to see this. Our cells can last up to 140 days in the freezer without loss in sensitivity. And this in turn made our operation much more streamlined. We don't have to do everything in one batch. We can, time shift their operation into multiple batches and be able to care for the work-life balance in our team while still producing high-quality data. Our data is much more sensitive and much more homogeneous than that from other academic and industry teams. And last but not least, we have much lower batch effects in our data. I won't go into too much into this heat map, but suffice to say that a common way to analyze perturb-seq data is to cluster the perturbations based on the transcriptional outcome impact of these perturbations. And often you're able to cluster genes from the similar pathways together, and we see the same thing in our data. So just a very simple clustering analysis on our top perturbations readily group proteins from the same pathways together, for example, exosome complex, ribosomal biogenesis, and protein synthesis. Something that was quite unique in our data that we noticed is everything that I've shown you so far are bulk analysis, pseudo-bulk analysis, grouping all of the single cells, mapping to the same perturbation together for analyses. We haven't really looked into the single cell heterogeneity in our data, and we know not every cell is knocked down for their target gene to the same degree. Would we be able to harness that information and either filter out cells with weaker knockdown, only keeping the cells with penetrant knockdown for analyses? That'll clean up the data quite a bit. Or maybe we can even look at the cells with a weak knockdown and high knockdown to learn that those dependency biological effects of a particular gene perturbation. Well, to do that, ideally, you want to look at the target gene expression level, but that's not a reliable assay due to the sparsity of single-cell RNA-seq measurements. So we started to think about other proxy measurements we can do to infer the level of knockdown in the cell. Well, here we look at the guide RNA expression in these cells and reasoning that CRISPR machinery, the enzyme and the guide RNA, may be rate limiting in our case for knockdown. And indeed, we saw a monotonic relationship between the guide RNA that's present in the cell as well as the on-target knockdown in the cell. We can then harness this information to stratify cells with high guide RNA UMI and low guide RNA UMI, and in fact, they reflect cells with high on-target knockdown and low on-target knockdown. So that's quite useful. We can stratify cells that way. And also, if we look at the secondary biological effects, looking at the energy distance, how profound is the effect size of the downstream perturbational effects upon a knockdown. Again, the high guide RNA UMIs have much greater effects than the low guide RNA UMIs cells. So this shows that if your reaction is like us, where it's really limited by CRISPR amount and guide RNA amount, this might be a useful trick to help you stratify cells and learn additional information or just clean up your data. All of the analysis I show you so far are actually pretty challenging to do from a computational standpoint. For a small-scale perturb-seq, hundreds of genes, you can read the entire data into local memory and conduct analysis, no problem. But when we are talking about a genome-scale perturb-seq, each file from our screen can be hundreds of gigabytes, if not more. This is no longer easily possible to load into local memory. So we actually had to collaborate with NVIDIA and Foresight Labs to come up with a new analysis workflow that convert the conventional end data object into SC data loader file, which allowed us to slice and dice the data set and stream bits of the data into analysis. And this workflow is what made all of the previous plots possible. So everything I talked to you so far, the fixation industrialized data generation platform, which we call fixed perturb-seq, as well as the first two genome-wide perturb-seq data sets, which is at the time was the largest perturb-seq data release last June, were all published in a preprint last year. I'm very happy to report that the data has been downloaded, I believe, more than 100,000 times now. And we received positive coverage from industry news outlet, praises from key opinion leaders, Dr. Emma Lundberg from the Human Protein Atlas, and the data was made free for academic use. We really want to contribute to this field. We believe in open science, and we welcome commercial licensing. Again, very happy to report that the data has been executed and licensed by multiple commercial parties. Again, signaling interest in these type of causal data that are high quality, high volume for training models of biology. That was last June, and the Orion preprint was already the largest perturb-seq data release at the time. Fast forward half a year to the end of last year, we were completing the second installment in our X-ATLAS data generation, which we call Pisces. So Pisces is more than 3 times the size of Orion data. But more important than the data volume alone, it also has much more biological context, spanning 16 different cell types and exposures. And to make this happen, we actually had to continuously develop our perturbation platform to conduct some of the more complex experiments. And I'll walk you through some of that in a second. You might think that, when we can do 2 genome scale perturb-seqs, doing a third, a fourth, a fifth is just a walk in the park. Of course, biology is never trivial like that. So when we go into our hepatocyte screen, this is HepG2, a hepatocytic cell line. These cells behave what I would call like salmon roll or water balloon. If you handle them not with care, they pop, releasing their content and leaking out the RNA for further analyses. So we actually have to develop new workflows to treat them super gently and introduce live viable cell filtering steps so that we can preserve the best cell for downstream analyses. We also introduced iPSC cell screening, and here there's an additional challenge. iPSCs can be notorious for silencing transgenes, and we typically introduce our Cas9 enzyme stably into the cell. But if they get silenced over the course of iPSC differentiation, of course, it's not there to silence genes anymore. And so we had to introduce ways to knock in Cas9 downstream of a housekeeping gene, and therefore preventing it from silencing. And what you're seeing on the right here is a day 30 iPSC-derived cardiomyocyte. You can see it's beating just like a real heart. And the red color shows Cas9 gene expression is still very much active. So we have to solve for Cas9 transgene silencing to enable this experiment. Last but not least, we also conduct our first dual contact screen, where we take the T cell line, the Jurkat cells, and conduct the screening in both resting and stimulated state so that we can answer the machine learning question, can we train the model only on one of the states and ask it to generalize to an unseen state And I think that question you can only answer when you can generate these dual context or cross-context data sets. Perhaps the most ambitious screen that we conduct is an attempt to really maximize cell type with minimal experiments possible. So this is achieved by differentiating iPSCs or induced pluripotent stem cells using a media condition, apower media, that is undirected. So in this condition, iPSCs can differentiate to many different cell types out of the stem cell fate, so that we achieve a library on library screening regime where we conduct genome scale perturbations in as many as 10 different cell types all in one screen. To make this happen, this is very tricky because different cell types are can be fragile, more fragile than others. And so if we don't have a way to really preserve the state of the cell, you will deplete the more fragile cell types when you harvest for the more robust ones. So here we have to upgrade our platform from a older GEMX-based platform to a flex-based platform that involve formaldehyde fixation, which is a much harsher chemical fixative. And in turn, we have to modify every other steps downstream so that we can really chemically preserve all of the cells, harvest all of them without losing any of the cell types. And the end result is this 7 screening campaign totaling 16 different biological contexts, including cell types and exposures, and which culminated in the XLS Pisces. Again, as I said, it's more than 3 times the size of Orion, and it has all of these contexts. And importantly, this allowed us to test a few machine learning hypotheses. First of all, can we use the core data set spanning these 4 cell types to test a new machine learning architecture beyond X token prediction, which I'll talk more about in the next couple slides. Second of all, can we use these cross-context experiments to test the model's ability to generalize across different exposures for the same cell type? And last but not least, can we use a multi-cell type data set to test the ability for the model to generalize to an unseen cell type, a cell type that's held out from the training? But before we go to the machine learning models, just a couple of words about the data quality. I'll go through these pretty quickly. These are really some of the most highest quality data we've ever generated. These are, for those of you who are familiar with single-cell RNA-seq, these are routinely now pushing 30K UMIs detected per cell, 6 to 7K genes, unique genes detected every cell. We're really at the technical limitations of single-cell RNA-seq here, and very robust knockdown, 70, 80, 90% of knockdown across all of these cell types. The multi-context hard work really paid off. When we increase our data from 1 context to 2 to 3 to 4, all the way to 7, we really see that the data is able to capture more and more of known biology as defined by known protein complexes or known protein-protein interactions curated by databases such as ChIRP and StringDB. You do not see this increasing learning, increasing knowledge when you simply stack cell numbers within the same cell type. So if I just did one of the cell types 7 times, that data is not nearly as information rich as spreading that budget over multiple different biological contexts. That's harder work, but you learn much more nuanced biology from this rich context data set. How do we know that our perturbation is universally giving us phenotypes for us to learn? Well, we employed a very simple metric called a binary classifier. And the concept is very simple. For any given perturbation in a cell context, we mix perturbed cell together with a non-targeting control cell into the same pool. We then train a linear probe, a simple classifier, asking it to predict what cell is perturbed and which cells are non-targeting control cells. With the implication that the binary classifier fails at this task, that means either the perturbation doesn't do anything, or it does something, but that something, the phenotype, is invisible in RNA-seq. And by this simple heuristic, we can readily detect, especially in some of our later results, more than 50, 60, 70% of the perturbations across all of the expressed genome have a detectable phenotype. That tells us that CRISPR perturbation combined with single-cell RNA-seq is a broadly useful general assay to learn biology. Now, of course, not all perturbations, as you might imagine, are equally functional across all of these biological contexts. So how do we learn that? Well, we did a very simple analysis by clustering the F1 scores from the binary classification from the previous assay and cluster them across all of the 7 contexts. And the idea here is that if a perturbation is equally sensitive across all contexts, having high F1 scores across all 7, then this perturbation has a phenotype regardless of the cell type of interest. Some other perturbations, and you see these perturbations map to housekeeping pathways, which is exactly as you might expect. These are universally important biology, whichever cell type you do the experiment in. You see many other perturbations to be cell type dependent. And this is quite interesting. For example, if you look at this cluster, they have the strongest perturbation effects in iPSC cell types, stem cells. Of course, you see these perturbations are highly enriched in neural crest differentiation, genes that are involved in developmental biology. Of course, these genes are much more important in a stem cell context than in a terminally differentiated cancer cell line context. So this is an easy way to show that we capture both cell type invariant perturbations as well as context-dependent perturbations. Another way to really illustrate this concept is by taking the features used to classify these perturbations and mapping them in this UMAP. And so this UMAP now represents, every dot represents a perturbation from one of the 7 screening campaigns, colored by the cell type that the experiment was conducted in. You see that first and foremost, perturbations cluster into major groups of cells, mapping to a coherent cell type. Of course, a lot of the biology is driven by the context, by the cell type of the experiment. You also see that we see many perturbations deviating from their parental program and clustering into coherent biology, corresponding to specific pathways and complexes. And so this is, again, as expected. Now, something that's quite interesting is that many of these perturbation clusters, they are multicolor in origin, again, signifying that these complexes, they perform the same function regardless of the cell type they came from. One such example is the mitochondrial ribosome. You can imagine mitochondrial ribosome does very similar functions regardless of the cell type. So again, this is quite expected. Whereas other perturbation clusters, they are very context dependent. And so you see this is only the complex 1, which is important for oxidative phosphorylation, a process to derive energy for the cell. This is only detected in the 2 primary cell screening from iPSCs, not so much from the cancer cell lines. This is quite interesting, and this may be explained by the fact that cancer cell line may have found ways to rewire their metabolic programs to go around oxidative phosphorylation instead of relying on glycolysis for energy production in an effect known as the Warburg effect. And I would argue it's exactly these context-dependent perturbations that require us to put into effort to map perturbation atlases across different biology, across different contexts. And these are exactly the signal that we need to feed into a machine learning model to learn to generalize to different biological contexts. Great, so I talk a lot about the data generation. Now I'll switch gear and talk about the modeling that we built on top of this data. So we trained on top of the Excel Atlas data, Pisces data sets that we introduced, we trained a virtual cell model, which we called Excel, that can help us predict perturbational effects and enable these downstream translational applications. The machine learning context, the question here is set up as the follows. Given a control cell transcriptome, given a perturbation, can the model predict what is going to be the transcriptome, the molecular state of the cell from that perturbation? And we're able to do this at the genome scale because we have genome scale perturbation data in training. So then this can be formulated as a training problem where the model is exposed to genome scale perturbations and be able to ask to infer in an unseen context what are the perturbational effects in the new cell type or new exposure context. There were 2 machine learning innovations that were introduced in the training of these models. And number 1, we introduced a new architecture going beyond next token prediction to really remove gene order that's quite arbitrary in RNA-seq and allow multi-step inference of cell states. And the second innovation is that we're able to make grounded prediction based on known priors, and we are able to introduce these prior knowledge via cross-attention mechanisms. And I'll go through them one by one. The first innovation is that we move beyond the next token prediction, the autoregressive language model that was pioneered by models such as SCGPT. And the limitation here is that this made a lot of sense for human language model where there are intrinsic order. And how I like coffee and coffee likes I mean very different things in human language. So the order matters. But in the case of RNA-seq, there's no intrinsic order in gene expression. And it doesn't make complete sense that the expression of the fifth gene only depend on the expression of the first four genes that come before it and not the genes after. This is made even worse by the sparsity of single-cell RNA-seq. Many of the genes, you can fail to detect them, and you get zeros in these very sparse sentences, if you will. And that makes prediction of an intermediate position gene very difficult if you don't have a lot of information to go by. And to me, it makes a lot of sense that we move into this diffusion language model, which is bidirectional and is multi-pass inference. And so this allowed every gene expression to be dependent on to gather information from all of the other gene expressions. And we go through this in multiple iterations. And you can see here a language model example where the order of finalization of the inference is not in order of the words anymore. It can really come up in any part of the sentence. And as a non-native English speaker, when I was this makes a lot of sense to me. I wasn't able to, when I first learned English, go one pass in one directional and do token-to-token inference or output. You really have to put keywords down, analyze the whole sentence again, update the keywords, and improve the grammar. And finally, this will be a beautiful paragraph. So, this diffusion language model makes a lot of intuitive sense to me. The other innovation that we introduced is this concept of a cross-attention injection of prior knowledge. And here, we recognize the fact that when we ask the model to do perturbation prediction, these perturbations should not be injected as simple gene IDs that does not have semantic meaning. We know a lot about what these perturbations mean. We know a lot about what these genes do. So why don't we introduce prior knowledge on these perturbation encoding? And this is done by introducing them through 6 different prior knowledge streams. We introduce LLM knowledge about what these genes are in the process of gene PT embedding. We understand in the protein language model level what these genes mean and how protein-protein are similar to each other through ESM2 embedding. We also understand protein-protein interaction through StringDB databases, as well as cancer dependency map, cell painting, perturbation datasets, as well as SGPT embedding. All of these are introduced to the inference time through cross-attention mechanisms so that we represent these genes in a meaningful manner. The end result is a model that performs really well. So here I want to show you a first piece of result where we trained Excel model on 4 of our core data sets and held out 200 perturbations from the iPSC and the HepG to the hepatocyte format. And we asked the model to make perturbation predictions in these 200 held out perturbations per cell type. We show that the multi-step iterative step of diffusion language model really paid off. Through more and more steps, we improve on our Pearson delta, a metric that predicts the effect size of the gene expression changes upon a perturbation, as well as MAE, the global transcriptional faithfulness to the ground truth. And we compare our model against 2 industry-leading models, Cell to Sentence from Google and State from Arc Institute, as well as the old SCGPT model, across a variety of metrics, including Pearson delta and MAE, across these 2 cell types, iPSC and HepG2. And you can see that across all metric, Excel really achieves SOTA performance, especially in metrics that matters a lot of our perturbation prediction, where you have to detect the changes that happen upon a perturbation. Excel model really outshines other models. And this is a fair comparison for state, for example, where it is also fine-tuned on our internal data. And you can imagine the performance gap will be even larger if state wasn't able to fine-tune on our internal data sets. So this is truly an architectural comparison. Excel model doesn't just outperform other models on our internal data. When we evaluate on external data, the gluconetic genetic perturbation data, as well as the cytokine perturbation data from PARS, a different data set, again, Excel model really achieves state-of-the-art performance across all metrics tested. We also try to apply XALICE, even though it's trained on genetic perturbation data on the chemical perturbation data set. So here we look into Tahoe data, which is a chemical perturbation data set generated by Tahoe Therapeutics. This is using a very different single-cell RNA-seq technology. So we had to do some test time adaptation by freezing the cross-attention mechanism and passing the control cell, just the control cells in and out, so that our model gets used to a different single-cell technology, so to speak. And when we test the zero-shot performance of our XL model compared to the state model, again, we found that the model performs much better, even on chemical perturbation data sets in both Pearson delta as well as MAE performance. You might ask at this point, what's making the model perform so well? Did the prior knowledge help? And if so, which prior knowledge that we injected using the cross-attention mechanism is really helpful here? We were curious in that question too. So we did an ablation experiment by permuting the gene ID at the time of inference for each of the stream of prior knowledge. So if you're able to permute the gene ID, that should render this particular prior knowledge useless for inference. And we look at the degradation in model performance when we permute or ablate each of the prior knowledge streams. Very interestingly, we found that dependent cancer dependency map and StringDB, this protein-protein interaction databases, had the largest impact on our model performance. This matched a lot to my biological intuition, quite honestly. And StringDB was recently found to be an important part of model performance by a different cross-model comparison paper published by GenBio as well. Now we see that some of the other prior knowledge streams don't have a lot of impact on the model performance. I don't think it necessarily means that these prior knowledge streams are not important. It could just mean that they're important, but when you ablate them but retain some of the other databases, the knowledge is still present in our prior knowledge, and therefore we don't see a major impact on model performance. We're able to, in the remaining of time, I'll go rather quickly, we're able to also evaluate the model's performance cross-context. So here we look at the JERCAT resting versus the JERCAT activated state, and we ask the model to predict what's going to happen in the activity state, blinded to that biological condition. We also ask a linear baseline to do the same, and you can see that the linear baseline really cannot differentiate known biology that's important for T-cell activation from the rest of the perturbations, whereas our XL model really can uniquely predict the effect of these perturbations from the rest of the general perturbations well. So it can generalize across biological context in this unseen exposure context. We also achieved high scaling factor for our model. So we trained a XL model Ultra from the 55 million parameter that's very similar to the model size of the SCGPT, almost by 100 factor into this almost 5 billion parameter model, which we call XL Ultra. And we found this beautiful scaling law. As you increase the training parameters, we continuously decrease the test loss. I think this is one of the first times that the field observed such beautiful scaling law up to this, I think one of the biggest virtual cell model in genetic perturbation prediction. This XL Ultra model has great performance. So we tested the model's ability to predict out of cell types by blinding the model to melanocyte progenitor cell type from the training data set and asking the model to predict perturbation effects across more than 1,300 perturbations. So really genome scale perturbation evaluation in this unseen cell type. And really you can see that across the board, the model achieves better performance than our smaller XL model, but also then some of the other models such as state model or perturbation mean out there across a variety of metrics. And last but not the least, we can even ask the model to predict out of zero data. So this is asking the XL Ultra to predict in a primary T cell perturb-seq published recently by the Alex Marson's lab. Beautiful paper. They use a different single-cell RNA-seq technology. So again, we use test time adaptation to learn this new technological differences. And again, we're able to predict in these primary cells, which are very difficult, takes a lot of know-how to do. XL Ultra can predict more accurately perturbation effects than our smaller XL model, as well as other industry-leading models such as state model across, again, multiple metrics. This big data set, XLS Pisces, as well as the world's largest virtual cell model, XL, was recently published in a preprint in March. Again, it received positive coverage by industry news outlet. And very soon, we're going to make a subset of the data and a subset of the model and code available for the academic community. Great, that's most of my talk. I want to quickly summarize here. I'm not going to read through everything here, but 2.5 years ago, we started with a simple hypothesis for a virtual cell model to achieve state-of-the-art causal biology perturbation prediction. We believe it needed causal data. And I think 2.5 years later, hopefully I've convinced you that the hypothesis borne out to be true. Really, we showed through all of this effort that by generating high quality, high context diversity, and high volume of perturbation data, causal data, we're able to train using the latest architecture, really state-of-the-art virtual cell model to do counterfactual perturbation prediction. And we displayed a beautiful scaling law here. Current focus in the company is to continue data generation, increasingly not in these proof of principle cell line studies, but in disease-relevant contexts. Think primary cells, iPSC-derived multicellular systems, organoids, and direct in vivo perturbations to go after specific disease questions that we care about, and to expand beyond the simple loss of function, single gene perturbations to increasingly other type of perturbations, and go beyond single-cell RNA seeking to other modalities as well. And last but not the least, mine what we have today, data and model for novel biological and translationally actionable insights to advance our pipeline efforts here at Xaira. So this obviously took a whole village to pull off. Most of the data that I share with you today were completed by Adam's team, including Lexi, Dan, and Nader. All of the part of automation work were done by Sue Hong, and computational biology work were done by our beautiful computational biology team led by Anne, and on perturb-seq analyses, and our genomics core really performed beautiful high-quality data generation for XLS efforts. Our model were done by our beautiful bio AI team. And last but not least, want to credit my partner in crime, Bowen, as well as the rest of Xaira leadership for all of these efforts. Again, picture for the company. Preprint can be found via this QR code. If you have additional questions, happy to answer them now, or feel free to reach out on X or LinkedIn. Happy to take any questions. Thank you for your attention.
Thank you, Chu, for this great talk. It was really great to learn more about Xaira and the technology that's being produced. So now I'll open it to questions. And if you could just raise your hand, and when I call on you, you can unmute yourself. I'll start off with a question, just more broadly touching on your role at Xaira. What does your day-to-day look like? And how have you navigated any challenges to working across the many teams that are at Xaira and the different locations during the scientific discovery process?
Great question. I think working in a company like Xaira, one of the most exciting things is that we have talents across very different disciplines, all working under the same roof. As you've already seen from this presentation, obviously, functional genomics experts, genomic experts need to work together with computational biology experts, but also machine learning AI experts, not to mention all of the drug developers in the company. Having all of these disciplinary talents in the company is very exciting. We learn from each other every day, but it also poses challenges. We need to be, for us to work effectively together, what will not work is if you simply generate the data, toss them over the fence and say, I'll see you 3 months later when you have a model that's trained on these data sets. We really need to speak each other's language and be able to cross-pollinate ideas. And that takes a lot of willingness, open-mindedness, and curiosity to learn from each other. And I think that's one thing that Xaira did really well. And so, if you are interested not only in furthering your own domain expertise, but also learn from other experts in the company, I think an environment like Xaira is great for doing this cross-functional work.
Yeah, that's great. I think getting that interdisciplinary perspective and breadth of knowledge is also always very important and very interesting. And now, I see Mohammed has raised his hand. Mohammed, if you want to ask a question.
Yeah, can you hear me?
Yes.
All right, thank you so much for joining us today and for this amazing talk. I want to preface this by saying I work in radiology, so this is maybe outside my field. But since data generation was a big part of the pipeline and the company, when do you think we will get to a point where we can use a trained virtual cell model to generate data, so that we can generate synthetic data to then further train models instead of having to do these large-scale data generation pipelines that are super expensive?
That's a fantastic question. Uh, Unlike several other fields, I think when you have physics rules of the world, you can start to generate physical data that's synthetically, right? That are high quality. Unfortunately, the rules of biology are pretty scarce. We have very few rules. Central dogma may be one of them. I don't think we're at a place where we can reliably, synthetically predict what's going to happen to a cell's program upon a perturbation. But the closest thing to that may be instead of perturbing in human, which obviously will not be scalable at all, perturbing in these in vitro avatars of human via these cellular models. That can be seen as a proxy to human perturbation experiments in many ways, and that we can generate causal data this way. And I think the value for a virtual cell model is that when we have enough these training data and model generalize well enough to unseen context, do we always have to run these very expensive, very time-consuming, high-throughput data generation campaigns? Or can we use the virtual cell model as an oracle to design better experiments? And we can do limited experiments that's informed by the model, not informed by human anymore in the lab, and use those data to come back and update the model in a lab in a loop fashion, and iteratively fill in the gap in the knowledge for the model and make it go better and better. Obviously, that's a super exciting paradigm to generate data on, but also solve a practical limitations in the lab because many of the experimental systems that I like to do experiments on are not amenable for high throughput screening. If I want to do experiments in mouse models for human disease, it's not easy to do 20,000 gene systematic genetic perturbations in all mice. If I want to do experiments in complex cellular models like organoids, I cannot easily do 20,000 genetic perturbations experiments in those. So having a model, having as an oracle to generate hypothesis, high quality hypothesis, so I can do the best experiments in the lab, I think we'll very excited to see that come to fruition in the near future.
And then we have a question in the chat from Kinga, asking, do you also plan to include other modalities such as proteomics, signalomics, and epigenetics? How are you thinking about translating the results from cancer cell line to primary cells and tissues?
A fantastic question. So two parts. On the first part, yes, my last slide already hinted at that. RNA-seq is great. It captures broad biology, but it's not the end-all be-all for all biology, obviously. Many assays beyond RNA-seq are already becoming scalable together with single-cell RNA-seq. Epigenetics is quite possible nowadays. Single-cell proteomics is quickly becoming more and more comprehensive. Other techniques such as mass spectrometry, as well as other lifestyle imaging are not quite there yet. So I think we'll stack assays that are scalable to these high-throughput screening campaigns, and use assays that are not quite as scalable in maybe these lab in the loop or fine-tuning regimes for data generation. So it's all about using the right modality, the right tool in the right context. To your question about extrapolating from cancer cells or cell lines in general to primary cells, that's indeed one of the exciting applications for virtual cell. And we hopefully show you a glimpse of that towards the end of my talk, where our model was trained mostly on cell lines and iPSCs. We're able to extrapolate that knowledge from that to the primary T cell screening from Marsden Lab. So they already show you some generalization capacity of these models. In general, I think there are a couple interesting scenarios I can see virtual cell add value to translational discovery. From primary cells that are easy to screen to primary cells is one of them, absolutely correctly. From simple monoculture systems to predict behavior of multicellular systems, co-cultures, organoids, I think that's another very interesting paradigm of model use. From one donor to predict effects in another donor, addressing patient-to-patient differences, I think that will be yet another very interesting generalization paradigm. From one assay, to your point, from one assay, single-cell RNA-seq, to other assays, proteins, live cell imaging, functional assays, will be yet another paradigm. And there we see some very interesting work coming out of the patient model side, right? GigaTime, for example, recently published by Microsoft Research. They're able to generalize from H&E, digital pathology, to spatial IF, spatial protein measurements. So that can be quite useful as well. So I'm very excited about all of these generalization paradigms and very excited to see where virtual cell will conquer first.
And I'm curious, are you also looking to expand to like single nucleus RNA data to like target like brain diseases as well?
Yeah, great question. I think single nuclear RNA for certain tissues is a necessary evil because it's not quite easy to dissociate cells while keeping the cytoplasmic content quite intact. So given the right tissue, we will definitely go there. I think what's quite exciting in the technological side is flex-based single-cell RNA-seq, because of the formaldehyde fixation nature, is now making it quite possible to generate full intact single-cell RNA-seq from FFP and formaldehyde fixed tissues and cell types. So we're seeing regular single-cell RNA-seq being more and more applicable to more sample inputs. But you're absolutely correct. For certain difficult to access tissue types, we may still have to go to single nuclear RNA-seq. And I think some of the literature that's being published suggests that that's actually still a very informative